The cell starts off in G0, a resting state where it waits for cues to start interphase, the stage in the cell cycle where it will prepare to divide. Now it enters the G1 phase. Here it will grow larger and copy its organelles. The G1 checkpoint checks the cell size, nutrient and energy levels, and checks for DNA damage before continuing division. Next, the cell will create a complete copy of the DNA in its nucleus in a process called the S phase.
One of the main errors that can occur during mitosis is cancer. This can happen when sister chromatids split and also during S phase of interphase when DNA is replicated.
The S phase is followed by the G2 phase. Similarly to G1, the cell grows and makes new proteins and organelles. Then the cell organizes its insides in preparation for mitosis. Right before mitosis is a G2 checkpoint, which checks for any mistakes in the DNA. This checkpoint marks the end of the interphase. Each checkpoint is controlled by both internal and external cues. The core regulators are proteins called cyclins and enzymes called CDKs. There are G1 cyclins, G1/S cyclins, S cyclins, and M cyclins. G1 cyclins remain at a steady level throughout interphase, but the other cyclins peak in their respective phases. These proteins will bind to the CDKs and direct them towards target proteins. Then the CDKs will either increase or decrease protein activity, controlling the checkpoints and progression of the interphase.
After the growth phase, we enter mitosis, which is when the cell divides into two daughter cells. The first step is prophase, where chromosomes condense and become visible. The nuclear envelope then breaks down, and spindle fibers connect to the chromosomes, which leads us into metaphase. The chromosomes now line up across the metaphase plate, getting ready to separate. We are now in anaphase. This is where the spindle fibers pull the sister chromatids of the chromosomes apart towards opposite poles. Once they reach opposite ends of the cell is ready for telophase and cytokinesis. Here, the nuclear envelope reforms around each set of chromosomes. The cell's chromosomes then return to their original, uncondensed state, and the cells divide.
Another cellular process involving the cell cycle is meiosis, in which 4 haploid gametes, or sex cells, are produced and prepared to fertilize into a zygote. Unlike mitosis, 2 rounds of meiosis will occur to ensure each sex cell receives a unique set of genetic material. During the first round of meiosis, the cell will undergo the same interphase process as in mitosis, with cell growth and DNA replication. However, differences will be first seen in prophase 1, where homologous chromosomes will cross over to exchange genetic material, resulting in the formation of tetrads. After this, the tetrads will line up on the cell's equator in metaphase 1, and homologous chromosomes are then split apart by spindle fibers during anaphase 1, unlike how sister chromatids split apart in mitosis. In telophase and cytokinesis 1, new cells are formed with 2 sister chromatids per chromosome in each cell. With the number of chromosomes already being halved in these new cells, they are considered haploid. These new cells then undergo meiosis 2, where processes are similar to mitosis, other than the fact that there's no DNA replication or major cell growth during interkinesis. However, meiosis 2 is still similar to mitosis in the aspect that prophase 1 begins with the nuclear envelope breaking down and spindle fibers being formed. Metaphase 2 similarly has sister chromatids line up on the equator, and anaphase 2 results in sister chromatids splitting to 2 polar ends of the cell.
Aneuploidy is fertilization of gametes under non-disjunction. Non-disjunction is when pairs of homologous chromosomes are unable to separate during meiosis 1. The result is one gamete receiving 2 of the same chromosomes, while the other gamete receives nothing.
Telophase and cytokinesis 2 then follow with new cells being formed. As a result of all of meiosis, each newly formed gamete receives 1 sister chromatid that has a unique cell of genetic material different from the parent cells.